Hello and welcome everyone to Life Science Across the Globe. My name is Janine Stevens and I am one of the Janelia organizers of this series. We are really excited for today's event on Origins of Humans and Culture, hosted by the National Center for Biological Sciences in India. We will have 1 hour of talks followed by a 15-minute moderated panel discussion and then 15 minutes for audience Q&A. And then immediately following the Q&A, we will have a special Meet the Panelists session. And we invite students, postdocs, and all other trainees to join us to stay on the call and ask questions of our panelists and hear from them on various career-related and mentoring-related topics. And now I'll turn it over to Professor Uma Ramakrishnan to make some opening remarks on behalf of our host, NCBS. Uma.
Hi, everyone, and welcome to this set of talks today. I'm Uma, and I'm at the National Center for Biological Sciences, NCBS, in Bangalore. So the National Center for Biological Sciences is a research institute under the Tata Institute of Fundamental Research. And at the NCBS, we have scientists who work across the scales of biology. And this results in really interesting science from ecology and evolution to cell biology, biophysics, biochemistry, and so on, as well as theoretical approaches to understanding biological systems. So I think it's really a pleasure for us, and we're very excited to host today's session. I think all of us are really curious about our origins. And in a sense, the study of human origins always includes study of human culture and is a very interdisciplinary field, bringing to bear genetics. The new kind of methods in genomics have really given a lot of push to our understanding in this area. So it's really exciting today to have a really great group of speakers. And to start off and introduce them, we have Professor Sarah Tishkoff from the University of Pennsylvania. She's a professor of biology and genetics there. And as someone who, when I was doing my PhD, she was someone who inspired me because she actually combined field work with genetics and genomics really early on to understand more about human origins. So with that, I'll turn it over to Sarah.
Thank you, Uma, for inviting me to participate in this really exciting symposium with such an excellent and diverse set of speakers. So my role today is going to be to act as a moderator. I'm going to be introducing each of the speakers. They'll speak for roughly 18 minutes, and it would be great if everybody could post your questions in the Q&A box. And we're going to hold the questions until after all the speakers have finished their presentations. We'll have then a general panel discussion, and I will address any of the questions that you've put into that Q&A box. So please do that. So I have the pleasure to introduce the first speaker, Dr. Himla Sujal, who is the Executive Officer of the Academy of Science of South Africa in South Africa. Himla, please take it from here.
Good morning, good afternoon, good day, wherever you are. Thank you very much, Sarah, for this welcome, and to the organizers for the invitation to participate. So let's start by sharing my screen. As I say, whenever we have to test it, it works. And then when it is showtime, we have another experience, but here we go, all set. Well, yeah, Sarah and I go back a long way. So I just want to share that with the audience. When she was still a graduate student in Ken Kids Lab, Sarah had engaged with us in South Africa. So it's very good to have rekindling of those fond memories. But today I want to feature a little bit more on my journey and experience doing this sort of work for over 3 decades until I changed my focus, and now I'm the Executive Officer at the Academy of Science of South Africa. And I thought in this presentation, what I would like to do is kind of juxtapose how our understanding of genes, genealogy, and history come together with a flavor of my experience from Africa. Well, no journey about our past is complete unless we understand the complexity of the narrative. No single discipline can converge on reconstruction of the past. And so what I would like to do is to kind of put into perspective the interdisciplinarity of the various fields, starting with history, linguistics, archaeology, anthropology, paleontology, then the genetics from population and evolutionary genetics, and also the advent of how all of this has played out with respect to medical genetics. And a few years ago in South Africa, I had the privilege of being part of a symposium where we brought together various experts and scientists from the various disciplines to cross-pollinate our ideas. And I'm showing you the cover of the book I was privileged to edit, which was the first time in South Africa that we were actually talking to each other rather than speaking to members in our single discipline. And so this is part of that journey on how we kind of cross-pollinate and talk to our colleagues in other disciplines so that the pieces of the puzzle we each contribute from our own areas of expertise can come together to give us a bigger picture. Now, we are all familiar with this kind of cartoon representation of genomic inheritance. So I am an individual. I didn't fall from the sky, but sometimes I think I do, but I am part of a family unit. And family units are parts of extended family units. And then we are part of a ethnic or a culture. We're part of a community, which may be very localized. Then in the broader context, we're part of our national, our regional, continental, and then part of the global sphere. So this kind of journey transcends from a living individual all the way into the past, which is somewhat different to the paleontological approach of where they look at specimens found from the past and try to reconstruct the present. And of course, we have different types of genetic markers. We can talk about mitochondrial DNA passed through the exclusive matrilineal line. We talk about the Y chromosome that's passed from father to sons. And of course, every single strand or segment of DNA we have in the nucleus is a combination of what we get from both parents. Now, while this cartoon starts at the great-grandparental level, showing us clear sticks of representing chromosomes, of course, we know that all of our chromosomes represents recombinations of many, many years into the past. So I also want to put out the concept that there are no pure populations, because part of the kind of racism issues that we've heard in the past kind of work around the idea that we're part of these homogeneous pure populations. So let's forget that exists, that we are all products of mixed assemblages of genetic material that has passed down to us over the eons. And if I were to use this cartoon that Ken Kidd presented some years ago, and I call it the Smarties diagram of the world. When you look in the African continents, yeah, we have a full book, box of the different complements of the Smarties in the box, but only a subset of that left Africa to eventually spread across the entire globe, taking with it subsets of that genomic information from Africa. Whereas in Africa, this continuum of the different genetic structures have been evolving over the eons. So Africa has also been in a state of constant evolution, even though some individuals left Africa early on. And then to kind of put what we have learned as a collective together, I only have 18 minutes to do the presentation. And if you were to look at some of the genetic or the autosomal markers I've done in collaboration with my former PhD student, Karina Schliebusch, who now works with Matthias Jacobsen at Uppsala University. When we did this sort of analysis using SNP DNA analysis, we found that on one branch of the human tree were placed all the Khoi and San populations that we examined. And it took a considerable bit of time before we started to see substructure in other parts of Africa. And so from this, we can deduce that living Khoi and San populations have retained some of the most ancient signatures or genetic signatures found in the world. Well, if you go back 100,000 years, they were not known as Khoi and San. These sorts of demic units have come in more recently in our cultural representativity of them, taking into consideration either their lifestyle by way of subsistence or language. And so these sorts of ethnic identities have been structured along the way based on certain commonalities that we see across human populations today. Well, from here, we've learned a lot in Africa. We've learned about the structure or the patterns of genetic variation that are different in some essence from people living in the western part of Africa, East Africa, Central Africa, and the southern parts of Africa. We have learned about how certain genetic elements from Africa left Africa, but we've also had an input from other parts of the world into the African continent. So Africa has been the continent in a reasonable state of flux, having given off some of its genetic structures, but also received input from outside. And we have used the patterns of genetic variation within Africa, coupled with some of the historical, archaeological, and other narratives to talk about migrations in Africa that would have eventually contributed to the shaping of the regional patterns of variation we see across Africa. And in fact, some people have said that within Africa, there are like multiple types of activities going on, giving rise to the differences that we see. And yet, we also have lots of similarities. Now, I just want to move on to what this all means when we talk about the value of genomics in society. In every single part of the African continent, you will hear different narratives about the value of how they see what they understand about their origins and their narratives concerning their past with their present day. And no better place to explore this than in Southern Africa. And this is the part that since leaving the bench that I feel very passionate about, because now I'm seeing science in a much broader perspective in terms of what is the value of science to the average person on the street? How do we convey what we learn from all the high-powered science and laboratory and analytical work that we do? How does this make a difference to the person out there? And so that's a very exciting component of this new journey. I want to explore this by looking at the place of the San in the history of Africa and the world. Well, the San have over the years been subjected to really harsh experiences. In the early days, because of the excellent tracking experiences, they were taken as trackers by Namibia, which was then Southwest Africa, to use against forces from Angola and so forth. And when this army was disbanded, they were then moved to a place north of Kimberley, about 100 km north of Kimberley, at a place called Schmidstrupt. And they were put in tented facilities. There were two groups of San that lived there. And I mean, their conditions were not the best. And so, this battalion was disbanded, and from Schmidstrup, they were eventually moved to a place called Platfontein, closer to Kimberley. And this is where some of the people who had spent time in that army now reside. And that community has grown, and they've come into their own, living in a much more structured environment, but not without much hardship. And so given what we have learned about the San, particularly the kind of antiquity that they have in their genomes. Everyone who is doing genetics wants to study the sun. And we have heard about parachute scientists coming in, taking material and going away. And so this kind of war on the sun, and eventually they produce their own code of conduct with respect to conducting research among them towards the end of 2017. And all they ask for are simplistic issues. Respect us, come through the front door and ask us for permission to do what you want, rather than flying through the back door. Be honest with us, tell us what it is you want to do, and not come with one proxy and then end up doing something else. They expect justice and fairness. They want to deal with us with respect and care. And so far, I must confess that in my dealings with the various communities, it's been good, because I go out and I work with them and follow their rules and regulations to be able to conduct the work. So the process that they have put in place is really, really clear to us. And of course, it's taken a lot of time to be able to take the science into the communities, work for days on end before you even start to collect a sample, to ensure that you have the opportunity to converse with people. And this brings me to the next section of my story, and that's the oral histories. So with a company locally called Jive Media, I produced some, cartoon type tools to take around with me, so that people could understand from a very easy perspective some of the things that we were trying to do. And I'm just showing you a clips here. So of course, the first form of our history is through the oral traditions. All of us sit and listen to stories our parents, grandparents, and close relatives tell us about the past. And these are the fondest memories that we carry with us as we go on. And then as we become more interested in the topic, we may create our own little family trees or genealogies and see how far back it can take us. Well, we can't go back too many years with genealogy, but at least with the kinds of testing we do with ancestry testing and population genetic tools, we can learn a little bit more about ourselves. So having had the opportunity of intersecting with my science with the public at large, we were forced in my laboratory to conduct genetic ancestry tests, because people were listening to some of the things that we were talking about as we got papers published or at meetings. And so I was involved in a documentary called So Where Do We Come From?, featuring many profiled South Africans and average people. And here I am giving the late President Nelson Mandela his genetic ancestry test result. That alone was a goosebump moment. But of course, it was only when I got back to my laboratory did I realize, oh, wow, I actually had a moment of having to explain to President Nelson Mandela what his history was all about. And his mitochondrial line fell on the branch we commonly refer to as L0D. And so, as we know, there are many Khoi and San people whose mitochondrial lineage falls on that branch as well. And because of that, it was amazing. Whereas in the past, you would struggle to engage with groups to invite them to participate in your study. Suddenly, I had this overwhelming response by various communities across the country to come and speak to them about the genetic tools. They wanted to be tested. And I mean, to this day, even though I'm not in that profile, I'm still getting requests from people to be tested. So there could be reasons why they want this. And some of them are like, people want to use this knowledge of their history to engage with their communities and the young people in particular, to bring them back to that value system that we have around family, etc. As you can imagine, in South Africa, the vast majority of people live in abject poverty with squalid conditions and may take to drugs and other sorts of things. And so people, community leaders, see these sorts of initiatives as mechanisms of bringing people together and fostering the values of family, of history, and identity with respect to one's past. But at that point, I want to say that genetic ancestry does not equal identity. I mean, each of us has an identity that could be very, very different to what we carry as part of our ancestry. And that's another important fact to get out into the communities when we engage with them. So talking about society at large, I mean, as science, we just using the COVID pandemic as an example, where how much of information on science we have out there, how much of information we have about misinformation and fake news. I think we all have a responsibility as scientists to be creative in the ways in which we engage with the public at large. And talking about one's history and one's past is one way of growing that trust and encouraging people to value science. Remember, when you talk about evolution, you come up against another big barrier, and that is people's values based on creation, their faith, and their religion. And without being disrespectful, if you just give people opportunities to think about these as possibilities without like saying, oh, well, I don't trust what you believe in, etc. Instead of being confrontational, be creative and sell the power and the value of science. That's been a very good challenge for myself. And it's been also a success story of being able to tell stories in a way that people can identify with. Last but not least, I just want to pick up on how we as scientists, irrespective of what we work on, dealing with the public is one issue, but dealing with policymakers is another. And so I would like to end by saying that we need to bring together these values of how we take science in terms of spreading that value across to one and all in the general public and also to policymakers, so that at the end of the day, what we value as scientists can be implemented. Thank you very much.
Thank you, Himla, for that excellent thought-provoking presentation. I just want to remind the audience that we're going to save the questions for after all the speakers have given their presentations. So please be putting your questions into the Q&A box. So I now have the honor to introduce our next speaker, Dr. K. Thangaraj, Chief Scientist at the Center for Cellular and Molecular Biology and Director of the Center for DNA Fingerprinting and Diagnostics in India. Thangaraj, please take it away.
So when you talk about India, South Asia, it's very, very unique because we have a huge amount of diversity exists in the population. Even without doing any genetics, by looking at their feature, phenotypic feature, one can tell about the population in general. There are population in India who typically look like Africans, as you see here, right? And there are people who are with a very light skin color, look like Europeans. There are people who are with the features of Southeast Asia. And there are people who doesn't appear like anybody, but they are very, very unique to India. So that's the diversity we have in India and South Asia as a general. So what is unique again in India or South Asia is that the populations are very well stratified in many ways. So one of the stratification is based on the language. There are several language groups, particularly there are 4 major linguistic groups. One is the Dravidians, people who live in southern part of India who speak Dravidian language. People in this particular area who speak Austroasiatic language. And the third one, Tibeto-Burman, people live in northeast and northern part of India speak Tibeto-Burman language. And the majority of the people who are in the north speak Indo-European language. These are the 4 major linguistic groups. Again, under each linguistic group, there are several languages. In addition to this broad linguistic group, there are several isolated languages, like the Andamanis. Within Andamanis, each one of them speak their own language, like Onge speak different language compared to Great Andamanis and others. There is another language called Nihalis. These are all isolated languages. Under each linguistic group, again, we have large number of social structures. A social structure in the sense there is a caste system, there's a religious group. And if you see that in the under caste, we have subcaste, breeding isolates, exogamous unit. Again, same way in the tribes, you see the similar structure, religious group. What is even more interesting is that every single endogamous unit, they marry within the group. They don't marry outside. That is being practiced for last several thousands of years. So that makes every population unique because they acquire unique set of mutation in this process. So what makes these kind of uniqueness is that in India, we have large number of population groups, 4,635 anthropologically well-established population groups that includes caste, tribes, 532, 72 primitive tribes. Half of them still follow hunters and gatherers lifestyle. That means they go and hunt for food in the jungle. They don't do any job, including the agriculture. In terms of identifying how old the population is in India or South Asia, so one of the great source is the ancient DNA. But unfortunately, unlike many other countries, because of the climatic conditions, we don't have a very good archaeological source. So this is one of the excavations where they found only the tools which probably the modern human must have used, but they didn't find any biological remains. But only the success ancient DNA analysis that was led by David, who will be talking after me, and Vashant, and we are all part of it, and successfully sequenced this bone sample found in Harappa region. But that's not really telling about the origin of people in the Indian subcontinent, because this sample is not very old, around 3,000 to 4,000 BCE, but this sample consists of the ancestral South Indians and Iranian hunter-gatherers. Okay, but I'll not go into detail, but in terms of origin of people in India or South Asia, it's not giving much of information. But having population with very strong practice of endogamy, we felt that analyzing the genetic composition of a large number of Indian groups might tell us about the history, when did the modern human arrive, and so on. While doing this, we found the first modern human probably arrived in the Indian subcontinent is none other than the tribal populations of Andaman and Nicobar Islands. I don't know how many of you are aware, there's a population who typically look like Africans. And we, there are several islands, 572 islands, 4 tribal groups who typically look like Africans. 2 of them look like Southeast Asian groups. We collected the sample from Onge, Great Andamanis, and the saliva sample from another tribal groups, Jarwa, and we processed that initially with the mitochondrial genome analysis. I'm not going into details, Sir already mentioned about it. And what was interesting is that the mutation which we found in Andaman tribes are very unique, which has not been reported or not been found anywhere else in the world. And by calculating the mutation rate, and we came to the conclusion that the Andamanis are probably the first modern human who migrated out of Africa and taking southern coastal route, reached Andaman and Nicobar Islands about 65,000 years back. So this is the first genetic evidence suggesting that the first modern human who migrated out of Africa are the Andaman tribes. So this is where we collaborated, myself, Dr. Lalji Singh and David. From here on, we took different aspects of looking at the population history in India. With David, we looked at the nuclear genome marker, that's Affymetrix array 6.0, consisting of 1 million SNP markers. We carefully choose out of 20,000 samples representing Dravidians, Indo-European, Austroasiatic, Tibeto-Burman, and also so-called the social groups. And we analyzed And just give you very briefly that the analysis showed that there's a PCA showing that these are all different Indian groups. These are HapMap samples of European, African, and Chinese. This tells how complex the Indian populations are, while these three HapMap samples clustering very tightly, suggesting that genetically they are more homogeneous, whereas here every single population is very unique, but still there's a genetic affinities between these groups. In this process, we also found that while Andaman is migrating, some group must have stayed in southern part of India, whom we call as ancestral South Indian. Later on, there was a migration towards north, and one group has come to northern part of India, and other group gone to Europe. And these two founding group, ancestral South Indian and ancestral North Indians, have admixed, gave rise to many populations. And to understand that when did this admixture must have took place, again, it took large number of population from south and north, again using the same markers. And we could establish that the ancestral South Indian and ancestral North Indians have initially gave rise to many population groups. And during the last 2,000 to 4,000 years, approximately, these two founding groups have admixed. And after that, last nearly 2,000 years, these populations started maintaining the endogamy. So they admixed, then the endogamy. And why the endogamy? Probably the caste system, which I insisted in the beginning, must have put more pressure on the population to maintain the endogamy marriage practice. In terms of the health and disease, look at what is the consequences of the endogamy. That's what we are going to see now. To understand that, we took again large number of populations within India and also in other South Asian countries, including Sri Lanka, Nepal, Pakistan, and many other countries. We used and found that there is a possibility of identifying large number of population-specific rare disease in this process, because large number of population in India and South Asia, they practice the endogamy marriage practice. Particularly, population in southern part of India, they follow the consanguinity, that means marrying within the families. And here the IBD score says that the large number of Indians, every line represent one population, are showing with high IBD score compared to the Ashkenazi Jewish and Finnish population that you all know that they are known for population specific, large number of population specific recessive disease. But unfortunately, we don't have any information about the population specific rare disease in India, except very few diseases. To give you idea about what is known in Ashkenazi Jewish and Finnish population, you can see that large number of diseases, most of them are autosomal recessive disease. Again, in Finnish population. How that happened? This is a pedigree. What you see here, this is a female, a male, married, and there are two daughters. And consider that there is a mutation here, particularly recessive mutation, which will cause the disease only when there's two copies of recessive mutations are there. So as long as that it exists in the heterozygous condition, no problem, because it's passing on generation after generation in a heterozygous condition. But after several generations, these two, now they actually are losing the track of their relationship, but they have come from a common ancestor. But now these two individuals, they think that they are unrelated, but they are in the same population. If they marry, carrying heterozygous mutation, that's when there's going to be a problem of transmitting both the recessive mutation, and when you have this kind of parents, there's a 1/4 of chance of getting homozygous mutation. Therefore, there's a disease, and that was confirmed by looking at there's a disease called progressive pseudo-rheumatoid dysplasia, where there are 30 patients who are come from southern part of India, carrying the mutation in a homozygous condition. The parents are absolutely normal, but they say that they are unrelated individuals. But when we looked at the flanking region mutation which causes the disease, 5 out of the 6 are having same haplotype, suggesting that they must have come from a common ancestor. So not only this, there are several other diseases. This is one of the example where the polymerase gamma, that's a nuclear gene, but it's essential for mitochondrial DNA replications. And if the mutation is existing in the heterozygous condition, the parents are absolutely fine even after 40 years. But look at this family, the consanguineous family, the individual with homozygous mutation are passing away or dying at very, very early age. These are, of course, there are some phenotypes which I want to go into detail. This is a non-consanguineous family, but the same population. You can see the parents are absolutely fine, heterozygous mutation, but the children are with the homozygous mutation and dying at very, very early age. So similarly, I think there are large number of such recessive mutations which leads to several diseases are existing in almost 1/3 of the Indian population or in South Asian populations. So that is going to be very, very challenging. We already started looking at some of this population sequencing and trying to identify the mutation which is responsible for that. So with that, I thank, there's a large number of people are involved in that. My own colleagues, right from Dr. Lalji Singh to my students, and David has been an excellent collaborator, and large number of this continues. Of course, the funding has come from different agencies. So with that, I thank for the opportunity. I hope I finish in time. Thank you very much.
Thank you so much, Thangaraj, for that fascinating talk. You did actually finish in time with even a couple of minutes left. And I just want to remind again the participants that, or I mean the audience, that if you have questions for the presenters, please put them into the Q&A box and we'll address those at the end. So I have the honor to introduce the next speaker, Dr. David Reich, who is professor of genetics and an investigator at HHMI from Harvard Medical School in the US. David, take it from here.
Good. Do you hear me?
Yes, yeah, we do. We could see your slides. Everything's good.
Great. So it's wonderful to be on this panel and with my colleagues and friends, and especially with Kay Thangaraj, who I've worked with now for almost 15 years very closely. I'm going to be talking to complement the previous talks about using genomic studies to understand origins of human and culture in the place that has been studied the best in the world, not because it's more important than other parts of the world, but just because that's where the most funding and the largest number of samples is. And it really shows some of the types of insights we can glean from this type of analysis. So I'm going to begin by showing a picture that is the type of picture that has often been shown in studies of human population history. Here's from a paper in 2012, which shows a picture of dispersals out of Africa after about 50,000 years ago of more complex and more deeply divergent population that formed within Africa earlier to that time and spread to different parts of the world. And there's a lot that's right about this picture in terms of the spread of people whose ancestry is closely related to Africans, and the great majority of ancestry of non-Africans does descend from this dispersal after about 50,000 years ago. But there's also a lot that's wrong about this picture, and we've learned a lot about what's wrong about that picture in the last really 5 or 10 years through application of ancient DNA and modern genomic techniques to show that this picture of initial arrival of people in different parts of the world and the people who live there today are descended in a simple way from the people who lived there before. There's problems with that picture. So frequency. So in 1960, Luca Cavalli Sforza, who's now 60 years ago, really started this field of using whole genome data to look at the past, made a bet that it would be possible to reconstruct the migrations of people around the world from variation in ancestry of people who live today, who's most closely related to each other. And the idea was that at some level, people would remain in the same place. And therefore, by seeing how closely related people are today, you might be able to make inferences about how people got to where they are today. And based on this, he looked at a set with his colleagues at a set of about 100 variations in protein polymorphisms. So these are like the ABO blood groups we often get measured for that vary in frequency across groups. And he saw which groups are most similar to each other with regard to their frequencies. And based on this, you can make maps of gradients of similarity and frequency. And this is a redrawing of a map that was first made by him and his colleagues in the 1970s, showing a primary gradient of variation in one part of the world in Europe, moving from Southeastern Europe to Northwestern Europe. And he and his colleagues interpreted this as different proportions of ancestry from something we know from the archaeological record, the spread of farming into Europe after 8,500 years ago from the Near East. And that this is sort of tracking the proportion of mixture. But now with ancient DNA from first farmers, we know that the proportion of farmer ancestry is actually almost the opposite of what's shown here. And it's going perpendicular to that, more from south or even southwest to northeast rather than the reverse. And the reason is that that spread of farming wasn't the only population change that transformed Europe. Other things did as well. So I'm going to be talking about this new scientific instrument that's come online in the last 12 years with the first whole genome data reported in 2010. It starts with a human remain, like a bone or a tooth. And in a clean room where the goal is to get beneath the surface of the remain to the parts that are not contaminated by archaeologists or people in the lab handling the material, we try to take out a particularly DNA-rich part of this bone or tooth. We extract a very smooth the protein, and we sequence in one of these sequencers that has dropped the cost of sequencing in 2 decades by a factor of about 1 million and has made it possible to carry out these studies. As a result of these innovations, there's been an explosion in the amount of data on a genome scale from ancient people. And it's now possible to regularly get DNA from people who lived 2,000 years ago, 5,000 years ago, 20,000 years ago, even sometimes more than 100,000 years ago, that from many perspectives is similar in quality to what you get from modern medical genetic sequencing. It's kind of a miracle that DNA is preserved like this, and it's opened up so many windows into the past. So from 2014, where we had about 50 individuals with whole genome data, we now have about 100 times that or more, and it's continuing to grow very rapidly. So what have we learned from this innovation? So what ancient DNA does is it allows you to get back beyond being trapped in the present and being only faced with the incredible diversity we have today. Incredible as it is, it doesn't capture the time dimension, and we're trapped in the present. So if you can look in the past and see how people from past archaeological cultures relate to other archaeological cultures, people who lived in those cultures, and to people living today, you can ask questions about how change occurs over time. So that's a very powerful thing to be able to do. So I'm going to start by zooming into Western Eurasia. As I said, not because this is a more important part of the world, but it's because we have so much data. And I'm going to start with the question about what a, quote, white person means. There's this idea that I think is very much around and that I basically learned when I was a kid from the school even, which is that people who are, quote, white today refer to a genetically or phenotypically broadly similar group of people living all the way from Central Asia and Iran to the Atlantic shores of Europe and have spread beyond through the history of colonialism in the last centuries and might reflect a migration into Europe and after about 50,000 years ago from a group that came earlier into Africa. But that's not true. So if you look at data from 2016, here's a paper we published that reported data from more than 8,000 years ago from the first farmers from the Near East and from Iran, and compared it to Eastern European and Western European hunter-gatherers that we and others had published. And what we found is that these four populations were each as different from each other as Europeans and East Asians. Even though today, this region, everybody's genetically pretty similar from the perspective of the frequencies of genetically variable positions, at that time, it contained four groups as different from each other as Europeans and East Asians. So if one was able to go back in a time machine and categorize people by their groupings, it would look nothing like today. So these bars show proportions of ancestry from these four sources. And the question immediately arises, which one of these is the primary ancestry of people living today? And how did the situation of relative homogeneity emerge from the situation of extreme heterogeneity? And the answer is that none of these groups disappeared. They just mixed with each other to produce the very low level of differentiation that we observe today. And so this phenomenon of, quote, white people, a relatively low degree of genetic differentiation, is just a product of profound mixture that happened in the Copper Age and Bronze Age of groups that were much more differentiated before. Now, this is not unusual in the world. We are increasingly seeing this as we get DNA data and ancient DNA data from many, many parts of the world, in Africa, in South Asia, in parts of the Americas, and in many other places. So now I'm going to zoom more tightly, not just into Western Eurasia, but into Europe. And in 2014, we knew that before 5,000 years ago, Europeans were a mixture of two ancestries shown in these bar plots for real individuals we had data from. Green is hunter-gatherers, and blue are these farmers from the Near East, from Anatolia, Turkey. But today, there's this big red ancestry that didn't exist 5,000 years ago. So something happened after 5,000 years ago in Europe that's really profound. And so we and others set out to try to understand that. And here's what happened. So I'm not going to go into the details of exactly how this analysis is computed because I don't have time. But what you see here are 1,000 dots corresponding to present-day people from the following locations in Europe and Western Eurasia. And they're colored by their locations. And this is the result of a principal component analysis. So the data is the following. We are looking at about 600,000 positions in the genome where people vary from each other. They're known as single nucleotide polymorphisms, where some people have one type and some people have another type. And since you have two chromosomes, one from your mother and one from your father, you can be two adenines, that's one of the DNA letters, an adenine and a cytosine, or two cytosines, so 0, 1, or 2. And so you have a 600,000 row table corresponding to all the positions analyzed and about 1,000 column table corresponding to all these individuals. You multiply the table by itself to see how closely related every other sample is to each other. You get 1,000 by 1,000 square table. And you perform principal component analysis to most efficiently separate the individuals. That's this plot here, which is an XY scatter plot, but I'm not showing the axes. And this is where the individuals fall, and you see an amazing pattern where these people split into two parallel gradients, Europe and the Near East, with relatively few people in between. The northern groups are at the top, the southern groups are at the bottom. I'm now going to gray out the modern individuals, and I'm going to show you how the ancient individuals fall. So if you look at where hunter-gatherers from Europe fall, they fall beyond Europe in the direction of European differentiation from the Near East. So what that's telling you, and we can show this and prove this, is that Europeans today are a mixture of these hunter-gatherers who no longer exist in unmixed form, but contribute in mixed form to Europeans and people from the Near East. Then after 8,500 years ago, when farming arrives in Europe, boom, there's this pileup of Anatolian farmers at the bottom of the European gradient. Meanwhile, in the steppes north of the Black and Caspian Sea, this group forms, but you still don't see people like Europeans today. That only happens after 5,000 years ago, between 5,000 and 4,000 years ago, and that mixture between this group and this group from the steppes and this group from Anatolia happens only after 5,000 years ago. So a summary is there's a first major migration of farmers from Anatolia into the Europe after 8,500 years ago. It's a profound event. It displaces about 80% of the local population and produces people with bar charts like this. And then there's another, and it wasn't known before ancient DNA, very large scale movement that in some places replaces 7% of the ancestry, like Germany, from people from the steppes north of the Black and Caspian Sea. So now I'm going to go and zoom into Britain, an even smaller place where we have quite amazing data. And I'm going to show you what happened as this steppe ancestry from the steppes north of the Black and Caspian Sea hit Britain. So this is a time transect from 6,000 years ago to 3,000 years ago. And here's the proportion of people who have ancestry from the steppe. And there's no people like that from 6,000 years ago when farming arrives to 4,500 years ago. The last big stones at Stonehenge, the big monument that some of you may have visited, go up and is built by descendants of these first farmers. And then after about 4,500 years ago, before 4,400 years ago, bang, there's a huge migration into Britain from the continent, and it's a 90% minimum population displacement from the continent. It's a very dramatic event. And ever afterward, there's large proportions of ancestry from north of the Black Sea in Britain. And the people of Stonehenge contribute very little to people in Britain today, whereas this is a primary ancestral population of people in Britain today. Now, just as a comparison, I'll show you what happened in Iberia. This is Spain and Portugal in the same time period. In the same time period from 6,000 to 4,500 years ago, there is no steppe ancestry. And at the same time, bang, it arrives. And there's actually a period of coexistence of these two populations where we observe individuals of both ancestral types. And then after about 4,000 years ago, these two groups collapse into each other with what's more like a 40% displacement of the local population. Not as dramatic as in Britain, but actually something quite dramatic happens. If you look at the coloring of the dots, that corresponds to the Y chromosomes of the individuals. The open circles are females and the colored circles are males, so we can determine whether their Y chromosome is typical of the steppe north of the Blackett Seas or not. And what you see is red is this steppe type Y chromosome. You see that after about 4,000 years ago, it's the only type of Y chromosome that's left in Iberia. So even though it's only 40% genome-wide replacement, it's about 100% Y chromosome replacement. And that's telling you that these incoming groups, the males from this group outcompeted the local males. And so it was probably a quite unequal interaction between the incomers and the locals, where the males coming in displaced the local males. So it tells you something about that event. Okay, now I'm going to go even more detail into Britain. What happened in Britain? So the Ice Age peaked between 25 to 17,000 years ago. The north of Europe, as the north of North America, was covered by a big ice sheet, and the areas just to the south of the ice sheet were also uninhabitable. Nobody lived in Britain at this time. There had been humans, like Neanderthals, for example, in Britain before this time, but they were completely cleared out by this climatic event. And then people start coming back into England at the end of the glacial maximum, first episodically and then to stay. And the oldest complete or nearly complete skeleton is someone individual found in Cheddar Gorge, where cheddar cheese is from, about 10,000 years ago. And genetically, you can actually predict what this person would have looked like based on their eye color and their skin color. And it's very clear that this individual would have had quite dark skin, almost as dark as many groups in Africa, or as dark as many groups in Africa, and blue eyes, a combination rare today, but frequent in hunter-gatherer in Europe. So how much ancestry from such people is left in Britain? That's what I'm going to show you now. So I'm going to tell you about what we know from the place in the world where we actually know the most about ancient DNA, because we have the largest amount of data. And so I'm going to start on the right with the percentage of ancestry from these hunter-gatherers like Cheddar Man about 10,000 years ago. So 10,000 years ago, it was 100%. 6,000 years ago, there's arrival of farmers, and it's a 99% replacement of the local population. So only 1 out of every 100 ancestors after that point would have been from the population of Cheddar Man. Then the event I told you about before, a 90% population replacement that results in now only 1 in 1,000 ancestors coming from that hunter-gatherer population. And then here's a new Samu work that we're working on, where we find a 50% population replacement in the late Bronze Age. Here's just to give you a flavor of it. Here's the proportion of farmer ancestry in Britain. And these are all individuals, these blue and red dots. And what you see is here, between 1,000 and 8,000 BC, that's 3,000 to 2,800 before present, there is a pulse and an increase in ancestry, and we can show that corresponds to a 50% population replacement. And then the Saxon is probably another 40% population replacement. So today, only 1 in 3,000 British ancestors lived with Cheddar Man, and only 1 in 30 or fewer in the population that built Stonehenge. We know this because we have so many British genomes, but it seems to be the rule in history that again and again, the people who live in a place today descend relatively little from the same people who lived in the same place long ago, although there are probably places that are more exceptional to the rules than others. But more and more, the more data we get, the more this seems to be what we observe. So I'm going to end with a riddle from ancient Athens, an ancient classical world. And it's also motivated by the question that I've been thinking about a lot, about what do DNA studies of history tell us about the relationship between genetics and identity and what you think of yourselves. And so the riddle is the following. So I'm going to quote Plutarch, who's a Roman historian and philosopher and essayist who wrote biographies of mythical and real characters. So he says, the ship where Theseus, this Greek hero in the youth of Athens, returned from Crete had 30 oars, and the Athenians preserved it because it was such an important ship. And they took away the old planks as they decayed over time, and they put in new and stronger timber in their places, insomuch that the ship became a standing example amongst the philosophers. People said, is it the same ship? One side holding that the ship remained the same, and the other contending that it was not. This is sort of related in my mind to the genetics, because over time, are British today the same as the British who built Stonehenge, or the British who are Cheddar Man? Thomas Hobbes, a philosopher in Britain in 1655, added another element to this ship of Theseus riddle. He said, if that ship of Theseus were, after all the planks changed, the same ship it was at the beginning, and if some person had kept the old planks and by putting them afterward together in the same order, he kept them in like a shack or something and again made a ship of them, then this would have been the same ship, right? And so there would have been two ships, the same. So the question is, like, what is identity? How much connection can people in Britain today claim to the builders of Stonehenge, from whom they are hardly descended at all? And my view is that people in Britain can claim a lot of connection to the people of Stonehenge. It's the place where they live today. It's a place they can justifiably feel proud of. But I think we can all feel connected to Stonehenge because we're all connected to each other. And I think we all have a shared right to our common past.
David, thank you for that outstanding, really fascinating presentation. I think now we're going to bring all the speakers back. So let's have them all join us. So far, I just see two of us. There we go. There's Himla. Hi, Himla. Just waiting for one more. There's Langaraj. Okay, welcome everyone for our discussion session. Boy, there was a lot. You left us a lot to talk about, and there's some great questions, but we'll just start with some really broad discussion. One of which is, all of you are either working with modern indigenous populations, or in the case of David, you're working with ancient DNA samples from people who are indigenous peoples in different regions. And Himla, you talked about some of the ways that this research can be done in an ethical manner, particularly amongst the San peoples in South Africa or Southern Africa. But of course, it's never one size fits all, so to speak, because there's so much cultural diversity. And even the San, there are San in Botswana and there's San in Namibia, and they may have different concerns. And how do you guys, I'd like to hear from each of you, how do you think we can go about doing the sort of research in an ethical manner? And how do we prevent, say, the misuse of the results that you find? Could those data be used to either promote discrimination, or in the case of Africa, we know a lot of the tribes are fighting over land use, you know, things like that. Is that going to make it worse? So let's just have, I'll go in order. Himla, you first, and we'll go in order of speakers.
Thanks, Sarah. So the most important thing is process. Working with the San, we know that the San Council, having only recently put out a code of conduct, have always had a mechanism in place in terms of obtaining a permit to conduct one's research, or if people want to film them, they have to apply for this permit. So we've always followed the due process. And in my instance, I even was invited to come and meet with the Sound Council in Kimberley, where they asked me a whole lot of questions, to which I answered. And from there on, I got the permit. And of course, we also have to get ethical clearance from the universities where we conduct our research. So when submitting that application, having a permit with the mode of operation and how we engage with the communities was helpful. How do we prevent misuse of information? I mean, we think of ourselves as responsible individuals, but when your data is published and out there, how it is used subsequently is outside of the control of the researcher. So we can only hope and assume that all people who work in this space follow the ethical codes of conduct that we all subscribe to when we apply for ethical clearance from our institutions. And one of the things I have done to kind of keep the link going with not just the SAN, but the communities among whom I've worked with, is going back and taking their genetic ancestry results to them and giving it to them on a one-on-one basis, which means, you know, more time in the field. But I think that's the way you show, you know, trust and you gain trust of people, that you just didn't come to us when you wanted something, but you also came to us when you got your results and you took the time to share it with us. And I think from there on, you build a relationship with the communities among whom you work. And that also adds to the demystifying of what science is and how it is that you can use science to empower people. Thank you.
Great. So sharing the results is a way of benefit sharing. Right?
Absolutely. Yes.
Yeah. Okay, Thangaraj, I welcome you to address this topic.
Yeah, by using the markers which we use for tracing the ancestry or understanding the relationship between the populations. So they're all neutral markers. They're not having any impact on their health and disease. So I don't think this can be anyway utilized no, for any other reason except for tracing the ethnicity of any individual. Of course, they're all published once we have the data, but I don't think that alone is going to be misused unless otherwise we use markers from coding region to understand whether so-and-so is susceptible for any disease or not. To do that anyway, we use case control study. I don't think that is going to available for the public. So in that sense, it's not no fear for anybody that it will be misused.
Okay, and David?
Thank you. I sort of agree with what you said earlier, Sarah and Himla and Thangaraj. It's extremely variable what engagement means and ideally should mean in different contexts. And it's, I think, a bad idea, maybe even a dangerous idea to try to impose a uniform framework for engagement and asking permission in different parts of the world. I think that in some places, there's very clear indigenous groups who have a stake in the work and need to be engaged. I think that may be the case in the SAN case often. In other cases, it's not so clear whether how engagement works in that way. For example, in Britain, how does engagement work with the local group? It's not clear it's the same situation. My feeling in this doing work in this area is that it's morally positive at the current moment in time to be inserting facts into the scientific literature and to be actually carrying out data that we try to collect in an ethical and serious and engaged way, publish it carefully, thoughtfully. And I think that constrains myth-making, xenophobia, and racism. I don't think it's always been the case that was the case. I think if some people were doing the type of research that we are doing and we were in Nazi Germany in the 30s or something like this, it's quite possible that our research would be more misused than used. But I think in this time, the insertion of facts into the scientific literature does more to constrain xenophobia, racism, and myth-making than to support it. I only sort of responded to one sort of set of things you said, but that's a thought.
So, David, I agree with much of what you say, but I just remind you about what's happening in the US right now and many places in the world and the rise of white nationalism. And it's going on today. And so people have sometimes misused, but results, but I do believe that it doesn't matter what you find in a way. They'll find something to support their beliefs.
I agree.
And, right?
So I just think that if we don't have science providing information about the past, all we have is a bunch of competing narratives. If we don't sort of have a belief that there's some type of objective way to try to understand how people are related to each other, all we have is what this person says and that person says. And if we at least have a scientific literature where it's a set of findings with people who you trust to analyze things responsibly, then you have to contend with that. And I think for white nationalism, like the thing I mentioned about white people being a very recent phenomenon and hugely mixed, about migration being the central theme of British history, for example, European history, and the history of many other places, about the idea of anybody being pure being really very, very fictional, that this is a shared feature of humanity. I think this is very hard to contend with for anybody, and it's actually very easy to understand. So I think that science is constraining these narratives, and if you throw away science and you just say it's what this person thinks and what that person thinks, sure, there's white nationalists, sure, there's nationalists of other types, but I think that science is a useful thing in constraining those debates and making it more difficult to make up those false narratives.
In fact, I can think of a great example where in the US, some of the white nationalist groups were trying to prove their so-called whiteness by whether or not they could drink milk. And they would give tests. If I can drink milk, then I am purely white. And then what they don't realize is that, as my group has shown and others, we found mutations in African pastoralists that allow them to drink milk. So African pastoralists can drink milk. And also what's interesting is that based on some of the ancient DNA work that your group has done, David, and others, it appears that these mutations associated with lactose tolerance may, tell me if I'm wrong, but it looks like they arose earlier in East African pastoralists than they even did in Europe, right? Wasn't that a pretty recent event? Or became common, at least became common anyhow.
Yeah, I don't think we have enough East African data to say that definitively, so I'm not sure about that at this point. I certainly think that the European lactase persistence variant rises to high frequency in Central Europe extremely late. So it's present, percolating at a very low level. The mutation arose, probably many thousands of years ago, but it's almost absent in the population and not really helping people to drink milk until the last, 1,500 years in a population way that matters.
Right. And now on the same sort of topic, I want to also discuss the issue of how does research on human genetic diversity, both modern people and ancient people, impact biological notions of race? And also, can this research be used either to promote inequalities or the opposite, to promote more equality? And anybody, I leave this open to anybody to answer.
I'll have a go. And first, I just want to respond to what David has just said. I mean, you're absolutely right, David, that ancestry and identity are two different things. And you've shown, precisely that. The issue is that in some parts of the world, like, for example, in Africa, where people's lived experiences, the impact of colonialism and the way in which people were treated, brings in that bit of emotion into the storyline. So you can present the data as you've done systematically and accurately and given us things to think about, how you assess what ancestry is all about. But unfortunately, when that emotion comes in through one's lived experience, it makes it difficult. And that is linked to the question that Sarah has just asked in terms of what is race. I mean, I actually preach the fact that we are all Africans, and that, if you go below the skin, we are all Africans because of this deepness in our genetic heritage. But very quickly, people will say, well, if I look at you and I look at the person next to me, I can see these differences. Don't be a moron. We are different. So how do you take this message of look beyond the skin and the hair about who we are in terms of our humanity and our common origins? And I typically do that using a tree as a metaphor, that you have different leaves harbored on different branches, but we all come down to a common trunk. And that is exactly what David has now demonstrated with his data, with the history of Britain. And so the more we try to bring the message to things that people can identify with, the easier it is to be convincing about, this race is one thing and racism is another issue. So that is another story that we need to unpack as well. Yeah.
Anybody else want to address that question?
Sir, I want to point out about the lactate persistent gene and the mutation. So in fact, that was found initially in Middle East, then spread to Europe. Probably from there, it must have gone to US, right? David, if you remember in European origin paper, in the bones which we did, which obtained from Luxembourg and other places, it was 8,000-year-old bone where the lactate persistence mutation, I don't think which was there in those samples, right? Yeah. So that clearly demonstrates around 5,000 years, but it's not too old.
Although at the same time, we have to be aware that we still haven't found all the mutations that are associated with the trait lactose tolerance. So there could be more out there and we're just missing them. So until we find them, we can't look for them in ancient samples. And I think that's also, just as an aside, a word of caution, in my opinion, about making inferences about past phenotypes, because David, you mentioned Cheddar Man, and I know that in all the images, Cheddar Man was really dark skin, but they didn't include at the time like mutations that we found that influence skin pigmentation and others that are being found now. So it can be really hard to make inferences about past phenotypes. I don't know if you want to comment on that.
Yeah, I mean, correction completely accepted. Especially, the particular skin lightening suite of genetic changes that is causes the pigmentation variation in Europe. It's probably semi-reasonable to think it's useful for predicting people 8,000 years ago in Europe. But if you project to people who live in different parts of Africa, either in the present or the past, that discovery of those variants, many of which arose outside of Africa or rose to high frequency out of Africa, are potentially quite irrelevant. And given, I'm just saying back to you what you know a million times better than I do, but they're potentially not relevant to the pigmentation variation in Africa, which is extreme. So just using one set of variants that are found in one place to predict others is not reliable. The question, you have to make some adjustment about how reliable polygenic or genetic predictors in one group are for another population, and that prediction of this Cheddar Man phenotype is a problem. I think it looks like our time is getting short. Is that right?
No, we have until 10:30. So pretty soon I'm going to be starting to transition to the Q&A from the audience. But continue if you have any other thoughts.
Yeah, no, I mean, in regard to your questions about genetic and race, if I could say just one thing, which is that it's challenging. I mean, race is not biology. It's not a biologically real thing, but it is the popular perception of biology. So it's an interesting thing. So it's people kind of intuitive folk understanding of the nature of differences amongst people that it's either imposed on them unwillingly from outsiders or people imagine about themselves. So it's not unlinked to biology because people's perceptions have some grounding reality, but it's often extremely wrong. Like in the United States, the racial category of Latino is very weird because it includes people of primarily African ancestry and people without any African ancestry at all, for example, people with primarily Native American ancestry and people with very little Native American ancestry. And it's more linked to language than to ancestry. So I think that it's complicated because the messaging that biologists and geneticists and others, anthropologists, give about whether race is real or not and what it means needs to be, people have sense that there actually is something there, that there are differences. And if you tell people there are no meaningful differences, a lot of people will disbelieve you because people's gut feeling is that there are differences, there are phenotypic differences. People are willing to be convinced that skin color is a superficial trait, but there are other differences that are meaningful. So I think that actually talking in a complex way, in a rich way, but also an easily understandable way about the nature of the things that Himla talked about, which are the time depth of human variation, or about Thangaraj talked about the time of arrival of people in different places. If you can communicate a sense of the depth of human variation, the nature of interbreeding and mixture between people, and how it is that this type of variation we perceive around us arrives, I think people will be able to contend in a more productive way with the intuitive sense of variation people see around themselves. And I think that's a gift that we can help to give. And if we don't stand up, if we don't take responsibility for helping to communicate that, I think we're failing in our responsibility as scientists. That's my view. So I think we do have to talk about it.
I totally share your perspective. Let's go ahead and move over to the audience Q&A. So Rahul has asked, and this is open to anybody, can you please simplify how is DNA sequence data used to trace and predict human migration routes? So we talk a lot about these ancient migrations. How do we actually do that?
Yeah, so if you want to look for the migration, there should be some genetic mutation which you expect that is present in one place, which is not there in another place. And you can also calculate the age of the mutation. And if, for example, the ancestral mutation, ancestral nucleotide and the mutant, then you expect that how many thousand years back this mutation would have taken place. If the mutation in one place is it's an ancestral allele, but in the other place it's a mutant. And if you calculate there is a 4,000 years that mutation must have took place, then obviously you know that this population must have migrated from there. This is just for a simple example I'm saying. So similarly, you have a large number of markers to do that. You use mitochondrial DNA markers, you use chromosome DNA marker, and you use autosomal DNA markers. So there's a software which can predict the age from a common ancestor, and you can able to address this question.
And following up on that, Himla, do you want to just touch briefly on what you learned from like mitochondrial DNA versus Y chromosome versus autosomal data?
Yeah, no, thanks very much. I mean, firstly, we need to understand the modes of transmission of different types of DNA so that the interpretations we make from those different data sets are taken contextually. So if I'm just using mitochondrial DNA, which is that element of DNA that's transmitted from mother to children, but only the daughters will pass it on. In the chat, I answered some questions about why mitochondrial DNA. And this DNA is transmitted in the absence of recombination. So we have a very exciting tool where we can study change over time in the absence of recombination, like we have in nuclear DNA, adding to patterns of variation. So mitochondria will tell us about how females have contributed in shaping the genetic profiles. Similarly, that non-recombining region of the Y chromosome that we use for study paternal transmission also has the equivalent in terms of building that story. And David has shown some examples of in his PC plot, how the difference in transmission by males and females have shaped things. Then we come to the autosomal markers now, and this is from the predominantly the nuclear DNA markers, where recombination and other aspects can also contribute to patterns of variation. And each has its limitations and the strengths. So if you're talking about the true history of a population, you obviously want to study all the genetic markers. So we need to be aware of the strengths and limitations of each when we tell the story. So what I would see, for example, with mitochondrial DNA may not necessarily be the same for Y chromosome, because males have certain differences in, for example, their mate choices, how far they would travel to find a partner, and so on. And then where genetic admixture comes into place with the mixing, as we've seen very nicely from both of the other two presentations, we see differences coming. So the bottom line is, when you're telling a story, accept and understand what the strengths and limitations of your marker system is, and then build story. At the end of the day, it's one history we're talking about. That's the human journey. So eventually there should be some degree of convergence of how we see, the total population history in context. So I think it is important that we understand that. And depending on the question that you wish to ask, you have a suite of genetic markers you can use at your disposal when trying to address these. Thank you.
Thank you for that excellent answer. Let me move on to a question by Shanti. She says, thank you speakers for these wonderful lectures and discussion. One of the issues that sometimes is a concern is the apparent differences between interpretations from the archaeological data and genetic studies, in particular as regards the timing of dispersal of modern humans into South Asia. This is perhaps owing to the rather sparse data from, or dates, I'm not sure, from Paleolithic sites in India. But an interesting issue to consider, especially in the light of a new research in terms of fossil finds, genetics, and archaeology. And to me, this also raises a general issue about, David, I'm hoping you can address, how is the sparsity of fossil material and the biases that we find material that we can get DNA from, ancient DNA in certain parts of the world, but it doesn't preserve tropical areas. And how is that shaping our interpretations of past population histories?
Yeah, Thangaraj can answer these questions as well as I can, but I'll take a shot. There are places in the world where our success at getting DNA that's old enough to be able to inform about population movements is incredibly poor. South Asia is one of them. Maybe it's the champion for them in some sense. It's very, very difficult in my experience. We have tried so many tribes and we have had a very small number of successes, but a huge number of failures. Sub-Saharan Africa is another, although not as bad, and there's many more successes, and it's much more so far. So because we don't have these anchor points of what ancestry was like at different time points in the past in South Asia more than 4,000 or 5,000 years ago, we really don't understand the ordering of arrival of different ancestral components that contribute to people today, or the possible existence of other ancestral components which are hardly represented today, in the same way that we do, for example, in places where we have much better data, like Europe and increasingly East Asia and the Americas. And so in Europe, we have evidence of, in the Ice Age, waves of mass movement across the region. And then those groups basically don't contribute very much to later groups, and they're replaced by other waves. There's no reason to think that that's not going on in other parts of the world. I mean, we have a paper in submission on foragers in East Africa right now, where we're doubling the time depth of DNA to 16,000 years ago. And it's quite clear that these groups are hardly connected to each other genetically. They're from the same deep sources, but there's very little what we call shared drift in the jargon, as if what you're seeing is just episodic expansions and blinking outs of populations in a highly diverse and humanly very, very complex landscape. And so without the dense sampling, you don't actually see that. And so you can get oversimplified pictures. Our picture of British population history before ancient DNA was similar than our history now, because we know that certain populations existed at certain times. So the more data we get, the more complex the picture that we observe. And I think you should be suspicious of simpler pictures. That's a sort of partial answer to your question. Completely agree.
And particularly in Africa, many regions, but we know this is a really complex demographic history. We're just seeing the people today. What about the people 5,000 years ago, 10,000, 20,000 years ago? They weren't just hanging out in one place. One thing about modern humans, we like to travel, we like to migrate, right, over long distances. And there's been a history of ancient migration events, even before modern humans, in terms of, archaic populations that were outside of Africa. A quick question I have then for David, I'm just curious, you may not have any thoughts on this, I don't know, but this recent dragon man, that it was a fossil found in China, 146,000 years old. And they say it's, not a modern human, it's something else. But then I'm wondering, is this Denisovan? Did they find Denisovan?
Yeah, this is an amazing skeletal find in Harbin in northeastern China. You should look it up on the internet if you haven't seen it already. It's an amazing thing.
It is.
And with terrible archaeological background, because it was hidden by a Chinese worker under Japanese occupation, and for 80 or 90 years or something like this. So it's like no archaeological context, but an incredible skeleton. And it's probably at least 140,000 years ago or something like that. And the skull volume is similar to that of Neanderthals or modern humans. And we now know from genetics and from ancient DNA that there are these widely dispersed groups of cousins of Neanderthals across Asia, which we call Denisovans, first identified from a finger bone and a tooth in Denisova Cave, and identified in mixed form in people of New Guinea and related populations in Oceania, indicating a highly diverse group of cousins of Neanderthals that was spread and diverse over hundreds of thousands of years of population separation in Asia. Because Neanderthals and modern humans both already have big brains, probably by this time, it's not surprising that there should be groups with big brains. And I would bet a modest amount of money that these are some form of Denisovans. The group disagrees with that. They argue that this is more closely related to modern humans than it is to Neanderthal, than it is. But I think that my guess would be just background Bayesian prior expectation these would be Denisovans. And I hope someone's doing more attempts on the DNA or these protein sequencing to try to identify whether these share diagnostic markers with Denisovans, Neanderthals, or modern humans.
That would be so cool. Okay, last question, because I want to make sure we have time. Thangaraj, there are a number of questions that kind of are asking similar themes, which is, for example, Li Fang says, when your team manages to identify the genes that contribute to hereditary diseases, do you think in a hypothetical situation, it could be used to inform newlyweds of their genetic compatibility and even avoid having children? Although it does sound quite terrible of being in a future where on top of prenups, now you have to get a genetic test. So I will point out that Ashkenazi Jews, certain groups do get genetic testing, and matchmakers will take that into account when trying to figure out who's a compatible match. But I'm wondering if you could address that, please.
Yeah, as you rightly pointed out, David also knows that it is being practiced among Ashkenazi Jewish. But here in India right now, it has not gone to that level. At least I'm happy that people are doing prenatal diagnostic in a family where there's an inherited disease. And right now, people are understanding the consequences of the disease and the mutations. And it's being allowed now to terminate the baby with the mutations. So that's, I think, it's the first step. People understand the consequences of mutations, and I hope that if it continues for next few years or maybe generations, people would adapt to that kind of situation where the Ashkenazi Jewish are practicing. There could be a premarital genetic counseling and testing. I'm sure that will happen soon.
Okay, thank you so much. This has been a fascinating discussion. I could just go on forever, but unfortunately we have run out of time. I want to thank all of the speakers. And now I'm going to pass things over to Janine Stevens.
Thanks so much, Sarah. Before we close out today, I just want to remind one more time the students and postdocs and any other trainees to go ahead and stay on the call. We're going to have a short, maybe 15 minutes or so, meet the panelists session where you'll get to learn a little bit more about all four of our panelists today and ask some questions that you may have. But before we get started with that, I just want to say thank you again, Himla, Thangaraj, David, and Sarah, for joining us today. I thought the talks were fantastic. The discussion was fantastic. We really enjoyed it. So thank you so much. Thank you to the audience for joining us. Please be sure to come back again for the next Life Science Across the Globe event on August 4th at 9 a.m. Eastern. And that one will focus on a cure for HIV and is being hosted by the Institute of Infectious Disease and Molecular Medicine at the University of Cape Town in South Africa. So we hope to see you again on August 4th. And at this time, I think we will take just a 1 or 2 minute bio break for everyone to just catch their breath and take a sip of water, and then we'll get started with the Meet the Panelists session. Thanks, everyone.